Hello, Summit Climbers. Mr. McKee here, and we're going to be talking about genetics today. So I want to begin by looking at the calendar and giving you kind of an overview of what we're going to be doing. So we'll talk about the learning targets here in a second, but you're watching the video here, and you can access this video from the calendar if you need to. Give it a rewatch. Going to have you do a practice assignment after the video. Involving Punnett squares and some of the basic concepts that I'm going to cover with you. And if you need help with some of the vocab that we've covered previously in this unit, then check out the link there. That's kind of the basics. And if you've forgotten any of that material and feel like you need to deep dive into it a little bit more, or maybe you just want a little extra help, please check out those video links that I've put there also. And as always, reach out if you have any questions for me. Okay, so going to be talking about genetics, and here's our learning targets. I can describe how traits are passed from parents to offspring, and I can use a Punnett square to predict the probability of a trait being passed to offspring. So I should mention kind of a caveat here. In this lesson, we're going to be focusing on organisms that reproduce sexually, not those asexual organisms like bacteria. We'll touch on those later on in the year. But today we're focusing on sexually reproducing organisms. And what that means in a nutshell is sexually reproducing organisms are organisms that are a mix of both parents. Okay? All right, moving on here, let's kick off things with a genetics joke for you. What was the geneticist's favorite pickup line? I wish I was Adam and Eve, then I could get paired up with you. So if you don't understand that joke, it means one of two things. Either it's not very funny, and that's probably the most likely, or you need to review some of our DNA structure content from earlier in the year. All right, and you can check that video out on the calendar if you need to. Okay, getting into it here. You will recall from earlier that this is what we call the central dogma of biology up here. That means this kind of explains how we work and how other organisms work in a nutshell. It all begins with the blueprints inside the nucleus of our cell, which are the DNA. And the DNA contains information that gets basically transferred from the DNA to proteins. And then in between we have the RNA. And the proteins, in turn, are what gives us our traits. And phenotype is the fancy word for traits. All right, so DNA makes RNA, which makes proteins, which gives us our traits. And that's true for pretty much all our traits that are inherited genetically. And the names of those processes are down here. We've learned that before, but that's not critical to what we're talking about today. All right. So talking about organization here and some key terms we're going to be discussing. So chromosomes are found inside the nucleus, okay, which is the control center of the cell, as we have learned. And chromosomes, if you zoom in on them real close, you'll see that they're actually made up of bundled up DNA, and this diagram does a pretty good job of showing that, okay? And we recognize the double helix there of the DNA. So a chromosome is basically just made up of DNA and some proteins kind of holding it together and a few other things in there that aren't really important to our discussion today. And if you zoom in even more on what a chromosome is, you'll see that it's got both a chromosome and DNA, that there is sections called genes, and genes make proteins, as we discussed earlier. So a gene is a section of DNA that makes a protein, a particular protein. All right? So we just talked about chromosomes, and now we're going to discuss that an organism gets one set of chromosomes from each parent, giving them two sets total in their body cells. So right here we're looking at human chromosomes. You'll notice that they all match up for the most part. So both of these chromosome number 1s right here are approximately the same size, And if you stained them, you'll see that they have the same staining pattern. That's something we may get the opportunity to do in class. The only exception for this is when you get down to chromosome number 23. And in the case of males, chromosome 23 does not have matching chromosomes, and we call those famously the X and the Y chromosomes. Those do match in females, however, who have two X chromosomes, all right? So we're looking at chromosomes, and as a reminder, these chromosomes here are made of DNA, and DNA is made of genes. So on each of these chromosomes, we have, on average, there's quite a bit of variation because you'll notice the chromosomes are different size, but somewhere around 1,000 genes on average per chromosome. In total, humans, at least according to the current science, and this tends to change as we discover more and more, but humans have about 20,000 different genes, and those genes control all of our traits to various degrees. Okay? And as a reminder, you get one set of chromosomes from one parent and the other set from another parent, giving you two sets in each of your cells. Okay, and you will recall again from earlier in the year that meiosis and fertilization are the two processes by which we get those sets of chromosomes from our parents. So a father produces one set of chromosomes and during meiosis, and those chromosomes, of course, are stored in a sperm cell, and a mother produces one set of chromosomes during meiosis, and those are stored in the egg cell, and then those come together in the process of fertilization and form that first cell that we all begin as, called a zygote. And a zygote is the first cell that has two sets of chromosomes. And then from there, the cells multiply through the process of mitosis and form a mature organism eventually. And all the cells in that organism, again, have two sets of chromosomes. Here is, in case you are interested, some other chromosome numbers of other organisms. We're going to be talking about pea plants here in a bit. Famous because Gregor Mendel, the father of genetics, did a lot of research with pea plants. So notice that they have 14 total. That means two sets of 7 chromosomes. And of course, each of those chromosomes carries a bunch of genes on it. I don't happen to know the number of genes that a pea plant has off the top of my head, but that's something you could certainly look up. So as I've said many times here, genes are found on chromosomes, but not all genes are identical. There are alternative versions of some genes, and these are called alleles. That's a very important word to know. And if you zoom in on alleles, what you'll find out is that their different alleles are very similar to each other in most cases, but they might just have a couple of differences. So we're looking at 2 chromosomes here, and this could be chromosome number 2 from humans, for example. And on one chromosome that came from, let's say, mom, and the other chromosome came from dad, on mom's chromosome we have a lowercase b version of an allele, and on dad's chromosome we have an uppercase version of an allele. So the way that those differ is in their nucleotide sequence. Again, check out the DNA video if you don't remember what a nucleotide is. But notice that the lowercase b allele has a couple nitrogen bases here that are different. And that might be enough of a difference to make a different protein, which in turn could make a different trait. All right? So when we're talking about alleles, we need to consider combinations, because it's the combination of alleles that gives an organism its trait. So for example, in pea plants, you're going to, in your assignment today, look at these 2 traits, actually all 3 of these traits, I think: seed shape, seed color, and then plant height. And there's a lot of different combinations for alleles that these pea plants can have, and these are the phenotypes that they're going to get from those various combinations. We're going to talk about how that works here in a moment. So dominant and recessive. Some alleles, it turns out, have the ability to hide other alleles when they're both present. So let's consider seed shape here, for example. Let's use a symbol for the round seed shape allele. We're going to go with R because it's always the dominant trait that gives us the letter that we use, usually the dominant trait. There's some exceptions to that. And for the recessive version of that trait, which in this case is wrinkled, we're going to use the same letters we used for the dominant trait, except we're going to use the lowercase version. Okay, so we're not going to use a W there. Because these are the same gene, but they are different alleles, different versions of the same gene. So we want to use the same letter when we're talking about the same gene. All right? And so what this means is if a pea plant has both the big R and the little r, the dominant allele is going to totally hide the recessive allele. And we're going to have no idea that that pea plant has this lowercase r unless we do a little bit of clever reasoning. Okay? So dominant alleles hide the presence of the lowercase alleles. So the only way that a pea plant can have wrinkled seed shape is to have two copies of the recessive allele. Little r, little r. All right? And there's some terms, some names for these different combinations of alleles. I won't read this all to you. You can check this out. But we're going to use these terms a lot: homozygous dominant, heterozygous, and homozygous recessive. So you'll want to study those a bit on your own, perhaps even pause the video and write them down. Okay, finally, if we want to predict the probability of an offspring having a particular trait, we're going to use this tool that you can see on the screen now called a Punnett square. So let's say we have two parents that are both heterozygous, meaning they have different alleles for a particular trait. And that means they're big T or little t in this case. So let's consider the mom plant. I should mention most plants don't have a sex. Most plants have female parts and male parts. So this could be the same plant actually that's reproducing here. That's oftentimes what happens. So the mom plant could give either its big T or its little t. It's a coin flip. It's a 50-50 chance. And again, remember this happens during meiosis when those alleles are passed on to offspring. So we're going to show both of those possibilities here. Same thing with dad. It could be the big T or the little t. So we're going to show both of those possibilities. And again, that would be during meiosis. And then the square in here, which I'll circle in red, this represents all the possible combinations in the zygote of the offspring. Okay? So, for example, if dad gives the big T and mom gives the big T, then the child is going to be homozygous dominant. If dad gives the big T and mom gives the recessive little t, then the child is going to be heterozygous. If dad gives the little t and mom gives the big T, again, that child is also going to be heterozygous. And then if both parents give the recessive allele, we're going to have a homozygous recessive offspring. Because of the way dominance works, 3 out of these 4, or 75% of the children, would be expected to be tall. And only 25% would be expected to be short. And that's sort of the basics of how the Punnett square works.